Alright, this video is to help you with the liquid chromatography lab. You do need to make sure you complete the table, so you'll need to calculate the W's, the V raves, the K primes for the red and the blue dye for each run, and then you will also need to calculate the alpha and the R. Use the example I did for you for the pre-lab to kind of help you, and if you get stuck, you're always welcome to come see me. Sorry, I'm having trouble getting this to go where we want it to. So you notice that here are the post-lab calculations. Again, some of those you will need to calculate an average for. I would definitely recommend reviewing those sample calculations, especially if you have not done those yet, to help you with that. In terms of the post-lab questions, you have 5 questions. It says, what is it meant by polarity of molecules? What causes differences in polarity? We've talked a lot about polarity in class. I think you should be okay with that. In discussing solubility, the rule like dissolves like is frequently used. What does this mean? An example of like dissolves like would be salt dissolving into water, but oil not dissolving into water. Think about the polarity of salt and water. Think about the polarity of vegetable oil and water. The oil cannot dissolve into the water, but the salt can dissolve into the water. Another example would be different types of alcohols. Ethanol and isopropanol would dissolve readily into one another, but gasoline would not dissolve readily into ethanol. Okay, if you need more help with that, I would Google like dissolves like. You're asked to draw the structural formula. That's the Lewis structure of isopropyl alcohol. Isopropyl consists of 3 carbons, 8 hydrogens, and 1 oxygen, C3H7OH. The 3 carbons are in the middle, and then the OH for the alcohol is coming off of the middle carbon. You can always Google it if you're really having trouble coming up with the structural formula for isopropyl alcohol, but what you should look at is this is how does it differ in polarity from water. We know what the polarity is of water, and I think you're probably going to see some similar features with isopropyl alcohol, but I think you'll also see some features that might cause it to be a little different. So some molecules can have hydrophilic and hydrophobic properties, aka your cell membranes. They have the hydrophobic outside, which makes it very difficult for ions or molecules to get into your cells without using channels or being carried through. But inside it's more of an aqueous solution, and so it's going to be more hydrophilic, and that's going to allow more polar substances to be able to move through. So hopefully that helps with that. For number 4, it says the resolution should be greater than 1. So basically, what was your resolution? Did you have good separation? Did they overlap, or were you able to get them to kind of separate? I know with the first run, y'all were having a lot of overlap, but I'm hoping with your second and third runs, you may have seen a little bit better results in terms of your separation. 5, in the step gradient separation, this is the second part of the lab. The 4 separate fractions were collected. How do these relate to the polarity of the column and of the eluting solvent? So as you were going through and adding the different combinations, you went from very polar to a very nonpolar eluting solvent. And so what were the characteristics of those components that were found in those separate fractions? How did they match up with the type of solvent that you were using? And for the ones that weren't coming out, why were they more willing to stay with the column? I hope that helps. If you need anything else, please don't hesitate to ask.